For each presentation of RIKEN's research infrastructure, we have invited 1 or 2 European commentators, each representing prominent European research infrastructure in the same field. It is a pleasure to invite Dr. Yann Hérault, Director of the Institut Clinique de la Souris in Strasbourg, Coordinator of PHENOMIN, French National Infrastructure for Mouse Phenogenomics, and Vice Chair of the International Mouse Phenotyping Consortium, IMPC, where RIKEN Bioresource Research Center is also participating. So, Dr. Hérault, please.
Thank you very much. Good morning and good afternoon to all from France. It's a great pleasure to be here today, and I would like to thank you for the invitation. It's really a great pleasure to be here today and to testify about the major important role of the RIKEN Bioresource Research Center, with which I've been working for more than 20 years, as you heard already. So as you heard very nicely by Professor Toshihiko Shiroishi, RBRC is a major research infrastructure for biology and medicine. They have a highly set of competent teams for generating, maintaining, analyzing, and distributing a unique large portfolio of bioresources, which are used by many different types of investigators from academia and industry. And this is really unique, as far as I know, around the world, to have this large portfolio of bioresource offered by one unique center. I would like also to stress that RBRC is also a major key player in the International Mouse Phenotyping Consortium, a world-class research infrastructure with 21 research institutions, which are devoted to identify the function of every protein coding gene in the mouse genome. Indeed, you may know that the entire genome of many different species has now been published, and whole genome sequencing is becoming relatively quick and cheap to complete. But as stressed before in another RIKEN Research Center, despite this advancement, the function of the majority of genes remains unknown, and not only the protein-coded genes, but also the non-coding part of the genome. The IMPC mission is to fill this knowledge gap and create a comprehensive catalog of mammalian gene function that will be freely available for researchers. And this is actually the case. It's really a concrete initiative for open science. As the future chair of the IMPC, I'm very honored that the RIKEN Bioresource Research Center is involved in this international cooperation effort. And I must stress that they have contributed a lot to this endeavor since 2014, with more than 2.3 million data points and many discussions and improvement of our standardized tests and for the making of the mouse model. It is of major importance to sustain such infrastructure where research activities are serving the community at large, and also such research activities that are pushing the limits of our knowledge. I hope that we will continue to have the opportunity, also as a leading European Center for Animal Research, to have a strong partnership and alliances through our common goal with RIKEN infrastructure and more particularly with the RBRC. I think we all aim to serve the whole scientific community with a long-term perspective, to achieve the comprehensive catalog of mammalian gene function, and to provide transformative insight into the genetic basis of disease that will impact upon clinical diagnosis and management, and ultimately prevent, detect, diagnose, and treat disease. And I would like to thank you for your attention.